Welcome to this lecture on specialized chromatographic methods. We've covered HPLC and GC, the two workhorse techniques, which together handle perhaps 90% of all analytical separations. Today, we're going to look at three techniques designed for specific analyte classes that HPLC and GC handle poorly or not at all. Ion chromatography for ionic species, gel permeation chromatography for polymers and large molecules, especially biomolecules, and affinity chromatography for biospecific purification. Each one of these techniques is built on the same fundamental principles of partition, selectivity, and resolution, but with distinct separation mechanisms. We have 6 major objectives today. The first 2 cover ion chromatography, then we have gel chromatography, and then the last 2 cover affinity chromatography and technique selection. There will be 2 worked examples as well at the end, and 6 practice problems. This is the outline of today's lecture, and we'll work through each one in order, and then we will do the practice problems as normal at the end. Ion chromatography, separating ionic species. Ion chromatography fills a critical gap that HPLC cannot cover. Inorganic ions like fluoride, chloride, nitrate, and sulfate are not retained on a C18 column because they're charged and hydrophilic. They also lack UV-absorbing chromophores, so UV detection does not work. Ion Chrom serves both problems by using ion exchange resins that retain ions based on charge interactions and conductivity detection that responds to any charged species. Look at the diagram on the right. The resin bead has fixed positive charges on the surface. Analyte anions compete with eluent anions for these signs. Ions with stronger affinity for the resin are retained longer. The typical selectivity order puts fluoride first and sulfate last, because sulfate is divalent and has a much stronger interaction with the resin. Where will you encounter IC? If you work in water treatment or environmental monitoring, IC is the standard method for anion analysis under EPA 300.0. If you work in semiconductor manufacturing, ultra-trace ion analysis in process water is done by IC. In pharmaceuticals, counter-ion analysis of salt forms uses IC. Suppressed conductivity detection. The suppressor is the key innovation that makes modern IC practical. The eluent in anion ion chrome is typically sodium carbonate and sodium bicarbonate, which are themselves highly conductive. Without a suppressor, the huge background conductivity would overwhelm the tiny signal from the analyte ions. The suppressor placed between the column and the detector is essentially a cation-anion exchange membrane. It exchanges sodium ions in the eluent for hydrogen ions. This converts sodium carbonate to carbonic acid, which is a weak acid with very low conductivity. But it converts sodium chloride to hydrochloric acid, which is a strong acid with very high conductivity. The result, the background drops dramatically while the analyte signal increases. This improves the signal-to-noise ratio by 10 to 100 times, given detection limits in the low parts per billion range for common anions. Modern electrolytic suppressors regenerate continuously and require no reagent replenishment. Here's our worked example for ion chrome. It shows ion chrome in its most common application, drinking water analysis. The municipal water plant runs this test daily. The calibration table shows external standard curves for 4 anions with excellent linearity. The limits of detection are in the low microgram per liter range thanks to the suppressor. The sample results show all 4 anions are well below the World Health Organization guidelines. Notice the method simplicity. A single injection gives 4 simultaneous results in 12 minutes. Before ion chrome existed, each anion required a separate wet chemistry method: titration for chloride, colorimetry for nitrate, turbidimetry for sulfate. IC replaced all of them with one instrument and one procedure. This is a compelling example of how instrumental methods drive efficiency in routine quality testing. GPC SEC separation by size. Gel permeation chromatography, also called size exclusion chromatography, operates on a completely different principle from all the other techniques we have discussed. There is no partitioning, no ion exchange, no affinity. Separation is based purely on molecular science. Have a look at the diagram. The column is packed with porous beads that have a controlled pore size distribution. When a mixture enters, large molecules cannot fit into the pores. They are excluded and travel only in the interstitial volume between beads, so they elute first at the void volume. Small molecules can enter all the pores and explore the entire bead volume, so they elute last. Mid-sized molecules partially penetrate the pores and elute between the extremes. Notice that the elution order is the reverse of what intuition might suggest. Big molecules come out first, small molecules come out last. There is no chemical interaction. In fact, any chemical interaction with the packing is a source of error. This makes GPC uniquely suited for determining the molecular weight distribution of polymers, which is one of the most important quality parameters in polymer manufacturing. GPC calibration and molecular weight averages. Gel permeation chromatography is calibrated using narrow molecular weight standards, typically polystyrene for organic solvents like THF or polyethylene glycol for aqueous systems. The calibration plot shows log molecular weight versus retention time, and it should be approximately linear over the useful separation range. Outside this range, molecules are either fully excluded from the pores or fully penetrated. The three molecular weight averages are critical for polymer characterization. Mn, the number average, treats every chain equally regardless of size. Mw, the weight average, gives more weight to larger chains. The ratio Mw over Mn is the polydispersity index, which measures how broad the molecular weight distribution is. A PDI of exactly 1.0 means every chain is the same length. This is essentially impossible except for biological macromolecules. Typical synthetic polymers have PDI values of 1.5 to 5, depending on the polymerization mechanism. The block at the bottom explains why PDI matters practically. Two polymer batches can have the same average molecular weight, but very different distributions, leading to different mechanical properties, different processability, and different performance. GPC is the only routine technique that reveals the full distribution. Worked example, GPC molecular weight determination. This worked example shows how GPC data is processed in a polymer QC lab. The chromatogram is divided into slices at regular retention time intervals. Each slice is assigned a molecular weight from the calibration curve. The detector response, represented here by the peak area at each slice, is proportional to the mass of polymer at that molecular weight. To calculate Mn, we sum the heights divided by the sum of heights over molecular weight. To calculate Mw, we sum heights times m squared divided by the sum of heights times m. The PDI of 2.82 tells us that this is a broadly distributed polymer consistent with a condensation polymerization process. Whether this batch passes depends on the specification. Here, a PDI limit of less than 3.0 is typical for a polymer resin, and the batch passes. In a real lab, the data system performs these calculations automatically. So you need to understand the underlying mathematics to interpret the results correctly and troubleshoot when they look wrong. Affinity chromatography, biospecific separation. Affinity chromatography is fundamentally different from all other chromatographic techniques. Instead of exploiting general physicochemical properties like polarity, charge, or size, it uses specific biological recognition events. The selectivity is extraordinary. You can purify a single protein from a crude cell lysate containing thousands of other proteins in a single step, achieving greater than 1,000-fold purification. The process works in 3 steps shown to the right. First, bind. The crude mixture is loaded onto a column containing an immobilized ligand. Only the target molecule recognizes and binds the ligand. Everything else flows through. Second, wash. Buffer is passed through to remove any loosely associated contaminants. Third, elute. The conditions are changed to disrupt specific interaction. This might be a pH change, a salt gradient, or addition of a competitive ligand. The target elutes as a concentrated, highly pure fraction. This is the standard first step in monoclonal antibody manufacturing, one of the largest sectors of the biopharmaceutical industry. Protein A affinity chromatography captures IgG antibodies with greater than 99% purity in a single step. Here we have a decision tree asking us about our chromatographic technique. The decision tree summarizes technique selection across the entire module. Start at the top. Is the analyte volatile and thermally stable? If yes, use GC for its unmatched resolution and speed. If it is ionic, if no, then we ask, is it ionic? If yes, use ion Chrom with conductivity detection. If the answer is no, you ask, do you need the molecular weight distribution? If you do, then GPC or SEC is the only technique that provides Mn, Mw, and PDI. If no, is there a biospecific binding partner? If yes, affinity chromatography gives the highest single-step purification. If none of these apply, HPLC is the default technique because of its unmatched versatility. The key point, HPLC is the default. You only deviate when the analyte has a property that HPLC handles poorly. Today's takeaways, 5 of them. 1, IC fills the gap for ionic species that HPLC cannot. 2, GPC provides the molecular weight distribution. 3, affinity chromatography exploits biological specificity for extreme purification. 4, each technique exists because HPLC and GC have blind spots. 5, technique selection is logical, not arbitrary. Next, we'll look at chromatogram interpretation and quantitation. Here are our practice problems. Any questions? Thank you for listening.